Bismillahirrahmanirrahim. We have already discussed in our previous lectures about carbohydrate chemistry and different natures of carbohydrates. We have already discussed the reactions of carbohydrates, right? And its application. Today we will discuss a little bit about the polysaccharides and its natures and the different structural differences of different polysaccharides. Okay. And before going into polysaccharides, we will discuss some important monosaccharides or D family aldoses, right? And you see here, these all structures are shown that these all are belong to the D family of aldoses. Why it's called a D? Because the highest stereo center, that is, suppose if you are talking about the D glucose in highest stereo center, that is carbon number 5, the hydroxy, if hydroxy towards right side, it is called a D glucose, right? It is called a D glucose. Similarly, if you are talking about this ribose or this D-arginose, why it's called a D? Because in highest stereo center, the first one is high stereogenic center is this one, second one is this one, and third one is this one. So the highest stereogenic center, that is carbon number 4 in arginose, the hydroxy towards right side. Therefore, this is called a D-arginose, right? So this D configuration is due to this hydroxy at highest chiral center. Suppose in D-glyceraldehyde there is only single chiral center, right? In which hydroxy towards configuration of hydroxy towards right side. Okay. Similarly, you see here, these all are the D family of aldoses. But here I try to mention that if you are talking about some aldotriose, that is D-glyceraldehyde. If we are discussing about aldotetrose, that is D-theriose and D-erythrose. Why it's called a D-erythrose and D-theriose? Erythrose is the terminology most frequently used in sugar chemistry, in which the hydroxys of these hydroxys are on the same side. The configuration of both hydroxys on the same side. Therefore, these are called erythrose. On the other hand, theriose, this terminology is most frequently used if these hydroxys configuration is opposite to each other, right? Therefore, these both terminologies, erythrose and theriose, are most frequently used since these hydroxys are configuration of these both hydroxys are opposite to each other. On the other hand, if we are talking about the aldopentoses, that is the 5 carbon containing sugars that all are isomers to each other, but only the difference is its configuration, that is D-ribose, D-arginose, D-xylose, and lysose. If you are talking about the D-ribose, all three hydroxys on the same side, while if you see the lysose, first 2 hydroxys configuration on left side, while the third hydroxy on right side. So only the difference of the position of configuration. If you are talking about the aldohexoses, these are hexoses containing 6 carbon containing sugars, and most important class of monosaccharides. The most important one is D-glucose. D-glucose, you see here the aldehydic functionality, the first carbon hydroxy configuration towards right side, third one on left side, while the rest 2 hydroxys towards right side, right? D-glucose and D-mannose are epimer to each other. These both D-glucose and D-mannose are epimers to each other. Why these both are epimers to each other? Why these both are epimers to each other? You see, epimer in which the configuration of only one carbon is changed. In carbon number 2, you see all other structural and formula are same, but the configuration at position number to the hydroxy on right side in D-glucose, while in D-mannose at position number 2, a hydroxy configuration towards left side. This is the only difference. Therefore, if the configuration at only one position is changed, that are called that compounds are epimers to each other. And this phenomena is called epimerization. I have already discussed in my previous lecture how glucose convert into mannose, right? So this process is also called epimerization. Similarly, you see here D-talose, D-allose, in which all hydroxys towards the same site. So these are the most important, biologically important aldoses of D family, right? And if you are talking about the some disaccharides, the most important one is stable sugar, which we all are having in our daily meal, right? That is sucrose, disaccharide. That is a class of oligosaccharide. Sucrose is a disaccharide formed from D-glucose and D-fructose. As you know that this when sucrose is a hydrolysis, it give us D-glucose and D-fructose, right? And we already know that if we discuss its glycosidic linkage, that is always connected with each other through 1,2 glycosidic linkage through alpha glycosidic linkage. Okay, 1,2 glycosidic linkage के जरिए ये एक दूसरे के साथ कनेक्ट होते हैं, right? In which one glucose and one fructose molecules are connected with each other through alpha 1,2 glycosidic linkage, right? Sucrose is one another thing. Sucrose is a non-reducing sugar because of its acetyl linkage and because of its acetyl linkage, right? Any question till here? Medicinally. Okay. Similarly, if you are talking about the maltose, maltose is the disaccharide of D-glucose having alpha linkage, right? As cellobiose, cellobiose is the disaccharide of D-glucose having a beta linkage, right? You see here, there is a beta linkage. If you are talking about different other polysaccharides, polysaccharides are classified into two types. One is homopolysaccharide and another is heteropolysaccharides, right? Homopolysaccharide means homopolysaccharides are polymers of single monosaccharide where, right? Homopolysaccharides are polymers of a single monosaccharide. On the other hand, heteropolysaccharide contain more than one type of monosaccharide, right? Whereas heteropolysaccharide contain more than one type of monosaccharide, right? A polysaccharide made up of only glucose units is called a glucan, right? This is there are basically two classes, that is homopolysaccharide and a heteropolysaccharide. Homopolysaccharide are polymers of single monosaccharide, and whereas heteropolysaccharide contain more than one type of monosaccharide. For example, one glucose is attached with one fructose and one fructose is attached with one glucose. A polysaccharide made up of only glucose unit is called a glucan, since it is a homopolysaccharide. But if all our glucose units are connected with each other and form a polysaccharide, it's called a glucan, right? So three important glucans are starch, glycogen, and cellulose. So these are important ones, right? Starch, if you are talking about the starch, as we know very well, the starch is the storage form of glucose in plants is called starch, right? The two forms of starch are amylose or amylopectin. Basically, this starch exists in two forms, that is amylose and amylopectin, right? Amylose consists typically of more than 1,000 D-glucopyranoside units connected by alpha linkage. So this is connected with each other 1,4 glycosidic linkage, but in alpha system, right? alpha-1,4 glycosidic linkage. So amylose consists typically more than 1000 glycopyronoside unit through alpha linkages. On the other hand, if you see the structure of amylose, amylose adopts a very compact helical structure like this one, while amylopectin is similar to amylose but has branching points after every 20 to 25 glucose units. This is the only difference. Amylopectin similar to amylose but has many branching points after every 20 to 25 glucose units, right? Like this one, you see after 20 to 25 units there is a one branch like this one, right? Through alpha-1,6 glycosidic linkage or any other system, right? So this is the main chain and this is the branch, right? So main chain is connected with 1,4 glycosidic linkage, while its branching is connected 1,6 alpha glycosidic linkage, right? So amylopectin, obviously it is similar with this amylose, but has branching points after every 20 to 25 glucose units. The most important polysaccharide is glycogen. Glycogen is the major carbohydrate storage molecules in animal, right? Glycogen is similar to amylopectin, right? It is very similar to amylopectin that have branches, no doubt, but glycogen is similar to amylopectin except that glycogen has far more branching as compared to amylopectin. Amylopectin से glycogen की branches जो होती हैं वो ज़्यादा होती हैं. Branching occurs after every 10 to 12 glucose units in glycogen, right? While if you see in amylopectin, where branching points after every 20 to 25 glucose units, but in glucose branching but in glycogen branching occurs after every 10 to 12 glucose units in glycogen, right? Glycogen is a very large polysaccharide. Why it is so much large? Why it was need to be large? The large size of glycogen prevents it from leaving the storage cells, right? The storage of 10 of thousands of glucose molecules into one molecule. एक ही molecule के अंदर इतने tens of thousands of glucose molecule के units एक glycogen के molecule के अंदर होते हैं, ठीक है? The storage of 10 of thousands of glucose molecules into one molecule greatly relieve the osmotic problem. इसकी वजह से क्या होता है? एक तो osmotic pressure या osmotic problems body में इसको control करता है for the storage cells. This would be caused by attempted storage of many individual glucose molecules, right? एक ही molecule के अंदर tens of thousands of molecule एक glycogen के molecule के अंदर अगर एडजस्ट कर जाएं तो इसकी वजह से ऑस्मोटिक प्रेशर या ऑस्मोटिक प्रॉब्लम के अंदर जो इश्यूज हैं वो कवर हो जाते हैं. और दूसरा, दिस वुड बी caused by attempted storage of many individual glucose molecules, right? The highly branched nature of glycogen allows hydrolytic enzymes to have many chain ends from which glucose molecules can be hydrolyzed, right? अब जो ये as we know that glycogen को हम as directly इस्तेमाल नहीं कर सकते हैं. ग्लाइकोजन दोबारा ग्लूकोज में इस्तेमाल होता है और ग्लूकोज इज द सोर्स ऑफ रेडी एनर्जी फॉर द बॉडी. हमें हम जो इस्तेमाल करते हैं अपनी एनर्जी के लिए वो ग्लूकोज है. बेसिकली ग्लूकोज कन्वर्ट होता है स्टोरेज की फॉर्म में मेजर कार्बोहाइड्रेट स्टोरेज मॉलिक्यूल इन एनिमल्स. ये स्टोरेज की फॉर्म में ग्लाइकोजन में चला जाता है जो कि एक पॉलीसैकेराइड है और ये हम इसको इस तरह इस्तेमाल नहीं कर सकते हैं. ये स्टोरेज में जाता है, फिर एक एंजाइम की मौजूदगी में ग्लाइकोजन से ग्लूकोज में कन्वर्ट होता है, ठीक है. सो ग्लूकोज इज द सोर्स ऑफ एनर्जी फॉर द बॉडी, राइट. Basically, long-chain fatty acids of triglycerols are used for long-term energy storage. So this storage, glycogen storage, is basically long-chain fatty acids of triglycerols are used for long-term energy storage in our body and in different animals, right? The third polysaccharide is cellulose. In cellulose, glucose units are joined by beta glycosidic linkage, right? In cellulose, glucose units are joined by beta glycosidic linkage. Cellulose chains are relatively straight, right? Cellulose की जो chains होती हैं वो relatively straight होती हैं as compared to glycogen, ठीक है, ये straight होती हैं. इसकी जरूरत क्यों पेश आई स्ट्रेट होने की? द लीनियर चेन्स ऑफ सेल्यूलोस, हाइड्रोजन बॉन्ड विद ईच अदर, इनके दरमियान हाइड्रोजन बॉन्ड होता है to give rigid insoluble fibers found in plant cell wall. As you know, cellulose is responsible to making the cell wall of plant. So the linear chains of cellulose hydrogen bonds with each other to give the rigidity in cell wall, and insoluble fibers found in cell wall is due to these cellulose molecules which are in linear chain that are connected with each other through hydrogen bond, right? The resulting sheets then stack on top of each other, right? Sheets की फॉर्म में एक दूसरे के ऊपर होते हैं. Why human not use cellulose? जो ग्लूकोज जो है इससे हासिल क्यों नहीं करते हैं, ये भी एक स्टोरेज है. द रीजन इज ह्यूमन लेक enzymes to cleave the beta linkage. Since it is connected with each other, these cellulose are connected with each other through 1,4 glycosidic linkage, but it is beta in nature, beta 1,4 glycosidic linkage. Human lacks enzymes to cleave the beta linkages in cellulose, and so cannot use cellulose as a source of glucose, right? We cannot use it as a source of glucose because we have no such enzymes in our body, and they break beta linkage of beta 1,4 glycosidic linkage, right? So the difference between cellulose, glycogen, and starch is starch has less branches than glycogen. Glycogen has more branches, and there are storage of 10 of thousands of glucose molecules into इतने बड़े मॉलिक्यूल्स होते हैं. एक ग्लाइकोजन के मॉलिक्यूल के अंदर टेंस ऑफ थाउजेंड्स ऑफ ग्लूकोज यूनिट्स इसके अंदर कवर होती हैं, ताकि ये इसलिए होती हैं ताकि ऑस्मोटिक जो इश्यूज हैं वो कवर हो सकें. जबकि सेलुलोस में ये स्ट्रेट चेन्स होती हैं, लीनियर चेन्स ऑफ सेलुलोस होती हैं. सेलुलोस आर रिलेटिवली स्ट्रेट. इसकी लीनियर चेन्स क्यों होती हैं? The linear chains of cellulose is due to our hydrogen bond with each other to give the rigidity and insoluble fibers found in cell wall, right? Because cellulose is responsible to making the cell wall in plant, right? Why human not get glucose as a source of energy from cellulose? Because human lack enzymes to cleave this beta linkage, right? In cellulose 1,4 beta linkage, therefore human cannot use it as a source of glucose molecule to get ATPs, right? So any question?